Welcome everyone to this month's webinar, Emerging Therapies, Focused Ultrasound and Stem Cells. My name is Donna Greening and I'm a Programs and Services Coordinator at Parkinson Canada. I know you're all eager to get started, but before we do, I want to go over a couple of housekeeping items before we start. We have enabled closed captioning for the benefit of our attendees who are hearing impaired. You can turn it on or off by clicking on the button at the bottom that says CC Live Transcript at the bottom of your screen. You can click on Show Subtitle or Hide Subtitle. Please note that the CC Live Transcript uses auto transcription, so there could be some spelling errors as we go along. Also, at the bottom of your screen, you'll see two buttons, one that says Q&A and one that says Chat. If you have any questions for any of our speakers today, it's important that you put them in the Q&A space. We will have some time for questions and answers at the end of our session. The chat window is there so that you can chat amongst yourselves, but if you do have any questions for any of the presenters, it's important that you put them in that chat window as they won't be able to see what's in the chat window. Sorry, you need to put them in the Q&A window. So let's get started. It's my pleasure to introduce you to Drs. Lorraine Kalia and Suneil Kalia. Dr. Lorraine Kalia is a scientist at the Toronto Western Research Institute and TAN Center for Research in Neurodegenerative Diseases. She is also a neurologist specializing in Parkinson's disease and related movement disorders at Toronto Western Hospital. She is an assistant professor in the Division of Neurology, Department of Medicine at the University of Toronto. Dr. Suneil Kalia is a scientist at Krembil Research Institute and assistant professor in neurosurgery at the University of Toronto. His research laboratory focuses on understanding molecular mechanisms of protein homeostasis in neurodegeneration and on establishing model systems to study protein function in Parkinson's disease. As a neurosurgeon, his clinical focus is on the surgical management of movement disorders, particularly Parkinson's disease. Welcome, Drs. Lorraine and Suneil Kalia.
Good afternoon, everyone, and thank you for having us here today. We'll be doing a tag team presentation on a topic of great interest, I think, to many people in the audience and very much to us, which is emerging therapies for Parkinson's disease. As you'll see, we'll go back and forth between the two. And just for those of you who may be curious, we are a husband and wife team, hence the joint last name. So we'll be talking about focused ultrasound therapy, one specific trial that we've been involved with, as well as a stem cell therapy trial. And these are both exciting areas of investigation ongoing at the Krembil Research Institute at the Toronto Western Hospital in collaboration with some very talented collaborators, both in Toronto as well as in the United States. So as a start, I'm going to just provide a little bit of background to why we even care about emerging therapies in Parkinson's disease and why I'm sure you care about emerging therapies in Parkinson's disease. And the first is to talk about what we actually have, which are symptomatic therapies. And I'm going to just try and delineate a little bit between what we mean when we talk about symptomatic therapies, because in some scenarios, we often just think about treatments and there are differentiations between, I think, what we have available and what we're really striving for the future. What I've listed here, I think, is familiar to probably everyone in the audience, which is the symptoms that can be accompanied by Parkinson's disease. Recognizing that not everybody with Parkinson's disease have all of these symptoms, I think that many people will appreciate or relate to much of what I have listed on this slide. Motor symptoms are the motor abnormalities that have been characterizing the disease since its very early descriptions centuries ago. And we use that really in terms of trying to diagnose people with Parkinson's disease. And hence why when you see your neurologist in the clinic, they'll want to see how you move, how you tap your fingers, how you tap your feet, how you walk, because we're really looking at the motor abnormalities. But what we also appreciate is Parkinson's disease is not strictly a motor dysfunction. disease, but it's also a condition that affects what we call non-motor symptoms, or everything else unrelated to mobility. Cognitive impairment and dementia being an important aspect that can be quite debilitating for people, but a variety of other features that can be experienced by people with Parkinson's disease, such as psychiatric manifestations, sleep problems, dysautonomia, under which falls constipation, which can be quite common and quite bothersome to people with Parkinson's disease after we've treated their motor symptoms. This is a bit of a diagram to illustrate the importance of how these symptoms translate in a person who has Parkinson's disease in their daily life and their ability. And so what's depicted here across the bottom is time that progresses from left to right. And then from the bottom to the top of this graph is degree of disability. And this initial triangle down here represents the non-motor symptoms that people experience due to Parkinson's disease. This triangle here represents the motor symptoms. And the reason why I've stacked them on top of each other is to demonstrate how cumulatively they together add to the disability that people experience with Parkinson's disease. In addition to that, there's this third triangle, which I'll talk about a little bit later, which is the complications that can be associated with the symptomatic therapies that we have right now, which in and of themselves also contribute to disability. And clearly, as the time and disease progresses, there's an increasing amount of disability, which of course we would like to try and slow or stop. Underneath all of this, and the cause for all of this, is a process that we call neurodegeneration, which I often will explain to patients as being just a big word to basically mean that brain cells die at a faster rate than normal. All of our brain cells die in adulthood, but in Parkinson's disease, certain brain cells die at a faster rate. And because these brain cells die increasingly over the course of the disease, as a result, we have these motor and these non-motor symptoms. So the current therapies that we have for Parkinson's disease, many of you are going to be familiar with, and a huge kind of bucket list of what we use for therapies fall under this bucket of pharmacological, or basically pills, some kind of drug. So levodopa is our workhorse. There's a variety of other related medications, which ultimately all try and increase the amount of dopamine that are in the brains of people with Parkinson's disease, because that's what is lacking. So the neurodegeneration, or the brain cells that get lost in Parkinson's disease, tend to be brain cells that make this chemical called dopamine. So we use strategies to actually try and replace the dopamine. Down here is a picture of Sinemet, which was one of the original versions of levodopa carbidopa. We know that there are other therapies that can be helpful. So non-pharmacological therapies such as exercise can in some ways actually increase dopamine. And then there are more advanced neurosurgical therapies that are standard of care and very good options for a subset of people with Parkinson's disease, where ultimately the brain is either stimulated with deep brain stimulation or ablated, meaning destroyed by strategies such as using electricity, or more recently using FUS, or focused ultrasound, to actually make small lesions, we call them, in the brain. And still, although different than the treatments that we use with pills, these therapies, we think, also in some way enhance the circuits that involve dopamine. And what I just depicted down here, this is a picture, a cartoon of deep brain stimulation. And this is the more newer technique called focused ultrasound that's used in an MRI machine to make these lesions in the brain. Limitations of the symptomatic therapies are several. If we had no limitations to these symptomatic therapies, we wouldn't be having this talk today, because we'd be happy with what we have so far, but we obviously want to do better. And treatment complications are a big aspect of the limitations that we have with symptomatic therapies. Some of this you may have first-hand experience with. So motor complications can happen over the course of the disease, where people start to notice fluctuations in that their medications work when they first take their pills, but before they take their next set of pills, they feel that there's wearing off of the benefit. dyskinesias, or the extra kind of dancing movements that can occur, are also a consequence of having levodopa or dopamine related medications when you're further along in your Parkinson's disease. There are some behavioral side effects that come with these medications. And then for people who go on to surgery, obviously these procedures can come with potential complications that any surgery could be associated with, such as infections or complications with the devices that need to be used in these surgeries. And one other important piece of the limitations of the symptomatic therapies that we have is that we do replace dopamine with the variety of mechanisms that we have, but in particular with pills. This administration of the way that you take the pills is obviously non-continuous, meaning that you take a pill, several hours later, you take another pill, several hours later, you take another pill. And so the way it's being delivered to the brain is in bouts of delivery each time you take a pill, and is non-continuous and is non-physiologic, meaning that's not exactly the way brain cells deliver or release dopamine. They do it in a different way than what we do with the administration of pills. So if we had ways to actually better mimic the way that dopamine would be released by our brain cells, this may also improve some of the limitations that come with symptomatic therapies. And with this, I'm going to pass it over to Suneil to talk about stem cell therapies, which is actually one potential way of replacing these dopamine producing brain cells and potentially providing delivery of dopamine in a more physiological way.
So, and that's what sparked interest in the field probably 2.5 decades ago. And at that time, the Parkinson's field was very different than it is now. We know much more about Parkinson's disease and much more about the symptoms that are important to patients. At that time, the focus in Parkinson's was largely around the motor symptoms that Lorraine mentioned. And as cell transplant technologies came online, the field basically thought that maybe Parkinson's is a great way, is a great target that we can use some of these new cell transplant technologies in, because the goal is exactly this: replacing the dopamine producing brain cells and treating the motor symptoms. So having a continuous source of dopamine, a continuous reliable source. And before I get into it, I think there's a lot of lingo and a lot of confusing terminology in the press and as you read about things. So I'm going to attempt to simplify it just so we can all be speaking the same language for today's presentation. So stem cells are basically cells that can divide, but the way you can look at them, they're like a blank slate. So a stem cell can, with special techniques, we call it programming with different growth factors, they can be pushed towards taking on certain characteristics. So in the case of Parkinson's disease, the field is really interested in taking stem cells and converting them into dopamine producing neurons with the goal of transplanting them. There's lots of different sources. So the work that I'm talking about from 25 years ago were probably more accurately referred to as cell transplantation, though that tissue was from fetal tissue, and that was transplanted into patients with Parkinson's. Fast forward to current approaches, and you can, from a blastocyst, you can derive embryonic stem cells that can be grown and propagated in the lab. So you don't have to have a fetal source of those cells anymore. And I'll talk about how they can be converted into dopamine cells with the goal of implanting them in patients. Another way is to deprogram, so taking a skin cell or a blood cell and deprogramming them and developing dopamine producing neurons and implanting them into a patient from a healthy donor, or you can eventually potentially take a patient's own skin cells, deprogram them to stem cells, and then reprogram them as dopamine cells, and then implant them back into the brain. So there are a lot of potential options. And from from 25 years ago, the technology has evolved quite a bit. So as I mentioned, 25 years ago, it'd be a fetal source of cells to transplant. Over the last 25 years, great strides have been made in the lab at understanding how to get stem cells growing in a dish, so you can have essentially an unlimited supply, and then manipulating those cells to take on properties of the dopamine producing brain cells that will need to be transplanted. So the challenges are many, but a lot of progress has been made over 2.5 decades. I do want to mention that this is still brain surgery. So at the end of the day, with current approaches, either 25 years ago or even in our current trials, we still have to do a brain operation to deliver the cells deep into the brain. The good news is we have, as neurosurgeons that specialize in this area, we have a lot of experience with doing this safely. And so our job is to deliver things in the brain. So either deep brain stimulation electrodes, or we can deliver cannulas or little needles into different targets very safely, and through those cannulas deliver the stem cell therapy. So it is important to just reiterate that it is brain surgery that's involved with delivering stem cells in its current state, as it was 25 years ago. So this was there were several trials done. So this field was established by first looking at preclinical animal models, and then cell transplantation was done in a handful of patients, and 2 big trials in the early 2000s were published. Unfortunately, those trials did not show a benefit compared to sham surgery in the larger group of patients, but for Parkinson's. But what it did show is that, number one, it's relatively safe approach, and it's technically feasible, and you can implant cells that survive in the patient's brain and do increase the amount of dopamine. This is a dopamine scan before surgery, and this is a heat map. You can see after surgery, there's more dopamine in the area of the brain where those dopamine producing cells are implanted. So this was very important work, even though the results were disappointing at the time. And it led to a lot of research to understand not just the feasibility, the safety, but what also happens to the cells. Now that we're over about 2.5 decades from the first patients being implanted, some have passed away from other causes, and scientists have studied their brains, and it turns out that the grafts can survive for 1.5 decades, 2 decades. What we've also learned, though, is that a brain with Parkinson's may pass on some of the Parkinson's like pathology to the transplant. But this is controversial, but it's good to know about, because it's a challenge that we'll have to overcome if this turns out to be true, is what happens to healthy dopamine cells when you implant them into an environment that's affected by Parkinson's. So that's an important learning over the last 2.5 decades. So where are we now? So the field is now, as I mentioned, moved away from cell transplants from fetal tissue and is growing cells in a dish. There's multiple different ways of going about this. I'm not going to go into the details. We're just painting broad brushstrokes here. Different sources, very complicated science that goes into these. So what I like to say is there's like a secret sauce, a concoction of different growth factors in the lab that are applied to the cells, different time points, to very precisely program these cells into dopamine producing neurons. And we have to study them. And so we're back at the stages of different clinical trials, but with stem cell transplantation, we're largely at the phase 1 stage, which is safety trials. And so this is a fairly current table showing ongoing stem cell work, and you can see multiple centers in the world that are involved in these trials at this point in time. And the types of cells that are used have slightly different sources, either coming from grown embryonic cells in a dish, or as I mentioned, from patients' own skin cells that can be reprogrammed. This is a busy slide, but I just wanted to include this to get across the idea that there's right now in the field, there are several studies that are ongoing. The source of cells is different depending on the study. The dosing is different. And then because some of these cells are not self or don't come from the patient themselves, one of the disadvantages of stem cells is that you have to be on immunosuppressives to prevent your body from rejecting the graft. And so that's a nuance that's very important, very specific to stem cell treatments. So far, very recently, one patient, actually 2 patients have been implanted in the world with derived cells from their own skin cells. And the results are just established that it's possible. This again shows that once this was done in this particular patient in the US, that they were able to produce more dopamine in the brain. And another large trial is underway, led by the team at Sloan Kettering. And this is a strategy of taking embryonic cells grown in a dish and using one of those secret sauces that I showed on a previous slide, and developing dopamine producing cells. A lot of work over the last decade has gone into establishing that these are safe. These cells are probably not toxic. They probably don't generate tumors. And we are about to be included at our Toronto site in a phase 1 trial with Blue Rock Therapeutics. So the PI here is Andres Lozano, and I'm a co-PI with Alfonso Fasano here. The eligibility criteria are very similar to the criteria for deep brain stimulation, but we are currently actively recruiting for this trial for stem cell transplantation, and hopefully the first patient will undergo this surgery at our site in Toronto next month. So there are a lot of challenges that remain. I'm not going to go through all of them, but there's a lot of work in terms of the cell source and survival of the cells. Another important thing, 25 years ago, DBS hadn't reached its prime time. Now any therapy has to compete against DBS. So we have to study that. Is stem cell therapy going to be a better symptomatic treatment than deep brain stimulation? And then I mentioned the other challenges with implanting cells. Even your own cells in the brain will be seen as foreign and potentially can be rejected. And then what about the brain affected by Parkinson's? How will this impact grafted cells? So a lot of active work in the field going on in this area. So I'm going to transfer it back to Lorraine, who's going to talk about disease-modifying therapies for Parkinson's.
So to pick up on our discussion about therapies, we talked at the beginning about symptomatic therapies. And now I want to switch gears and use this terminology that some of you may be familiar with, which is disease-modifying therapies. And for those who aren't, I'm going to just use a couple of visuals to try and explain the difference. So going back to this graph that I showed at the beginning with the symptoms that people have with Parkinson's, and then the neurodegeneration or those loss of brain cells that's occurring as a cause of the symptoms. We know up top here that we have symptomatic therapies that many of you are on that are trying to treat those symptoms. And ultimately what they're doing is they're hiding the symptoms. They're masking them. They're trying to alleviate them. But what they don't do is they actually don't change what's happening underneath, which is this ongoing loss of brain cells. And in that space is where we talk about disease-modifying therapies. And many of us feel that this is the holy grail in Parkinson's disease and other neurodegenerative diseases, meaning that we need to find these therapies that at the very least could slow the neurodegenerative process, and at the very best could actually stop the neurodegenerative process in its tracks. And that's a big area of interest for Suneil and I in the laboratory, and also a motivator for this next study that I'm going to talk to you about. I want to emphasize that obviously many of us who are studying Parkinson's disease or caring for people with Parkinson's disease care a lot about disease-modifying therapies. And this is just a diagram, probably out of date now by a year, of the different kinds of clinical trials that are going on. And the way that these are divided out here, these are the outermost circle are phase 1 trials, and then this innermost circle is phase 3 trials. The bottom here, you can see there's still a lot of work going on symptomatic therapies and making what we use for symptomatic therapies better. But you can also see up top here that there's a large number of different compounds being studied for their potential to actually stop the neurodegenerative process as disease-modifying therapies. So that's the good news. The bad news is that up until now, we have been trying to find these disease-modifying therapies, but none of the clinical trials up until now looking at disease-modifying therapies have provided any therapy worthwhile moving forward to actually giving to patients. And I just give you some examples on the left. This is a short list, definitely not a complete list, but just a short list of some of the more recent drugs, such as venglustat and this antibody up here that we had a lot of optimism for, showed a lot of promise in the laboratory and early studies in humans, but when actually tested to see if it slows the progression of the disease, did not show that in clinical trials. And always with these failed trials, we try and understand why that might have been the case. And there's a lot of speculation and a lot of reasons why this might be, and there's probably a combination of these that lead to that, whether or not we're making drugs that are affecting the right parts of the cell, whether or not what we're doing in the lab is actually modeling the disease properly, whether the clinical trials that we have aren't actually able to measure the slowing of disease progress because of the way that they're designed. There's always a question of whether or not we should be treating far earlier to be able to see benefits. And then there's this concept of biomarkers where we can actually measure. We don't have ways to really measure if the treatments are doing what we think they are. And then there's this other important aspect of the human nervous system, which is the blood-brain barrier, which we think might also be important for why some of these drugs didn't actually work. And that's where I'm going to just quickly switch for the last 5 minutes to talk about focused ultrasound therapy. I mentioned earlier on that focused ultrasound can be used to make a lesion or to ablate the brain. What I'm going to talk about here is actually a different application for focused ultrasound therapy, which is to open up the blood-brain barrier. And I'll just highlight here that this is work that we have been doing in collaboration with the team at Sunnybrook Health Sciences Center, led by Nir Lipsman and Heinonen, who are really leaders in the field of this technology. So for those who aren't familiar with the blood-brain barrier, or the BBB that some of us call it, it is actually a physical network that's composed of blood vessels and tissue, and is really a group of cells that are closely spaced together. And the purpose of these cells is to protect the brain. We don't want toxins getting to the brain. We don't want viruses or infections getting to the brain. And so we evolved this blood-brain barrier to basically protect the brain, which being partly biased, I view as the most important organ of our body. And while the blood-brain barrier is helpful and necessary to keep our brain safe, it can become a detriment when we're trying to develop therapeutics. So this is just a very simple cartoon. This is brain tissue where, for example, we want to get a drug. This is a blood vessel where drugs can be delivered. If you give a drug through the intravenous, your drug will be in your blood. If you take a pill by mouth, it eventually gets absorbed from the gut into your blood. And so for treatment purposes, we want things from the blood vessel to actually be able to cross this dotted line, which is the blood-brain barrier, and get into the brain tissue to try and prevent those brain cells from dying. And there are certain things that definitely don't like to cross the blood-brain barrier. So small molecules that are larger in size, biologics, we talk about these kinds of therapies that aren't actually chemicals, but are different kinds of biological materials. So DNA or RNA, they don't tend to like to cross the blood-brain barrier. And then larger antibodies or proteins. And then, as Suneil mentioned, stem cells, for example, they can't be delivered by the IV. They have to actually be injected into the brain because they won't cross the blood-brain barrier either. And one way to overcome the blood-brain barrier, as I just mentioned, that Suneil had mentioned using the slide that he showed previously, is a neurosurgical procedure. So you could actually take a needle and put it through, make a hole in the skull and then pass it through into the brain and inject what you want to inject in there. But obviously this is an invasive, more invasive procedure. And so there's been some interesting developments using focused ultrasound, as I mentioned, being headed by Claire Vohinanen and Neil Lipsman up at Sunnybrook Health Sciences Center, where they use the same kind of device shown here. This is an MRI machine. This is a helmet by which ultrasound is directed to the brain that's put into this helmet. And you can use it for thermal ablation, as I mentioned, which is shown in this diagram here, where we're actually making an injury to the brain. But more interestingly for us is actually using it to open up the blood-brain barrier. And this is done by low frequency ultrasound, where small bubbles are injected into the bloodstream and then are allowed to vibrate because of a low frequency of ultrasound. And with that low frequency of ultrasound, that cell wall, those kind of closely abutted cells that I told you were the blood-brain barrier, very briefly separate and allow some opening so that drugs can actually pass from the blood vessel out into the brain tissue. Dr. Lipsman and Dr. Heinonen and their team have shown this to be safe and effective in neurodegenerative diseases. They started by looking at Alzheimer's disease and also looked at patients with ALS or Lou Gehrig disease, and have found that you can actually do this in individuals and that it's reversible. So this is an individual who has Alzheimer's disease. This is an MRI of their brain. This little dot here shows dye after they'd opened up the blood-brain barrier, showing that actually dye could get into the brain, and then that afterwards, that opening actually closes and dye no longer goes in. They had approached us because they were interested in applying this to Parkinson's disease, and we were very excited to actually talk and work together to design a clinical trial to be able to apply this to Parkinson's disease, because we think it has a lot of promise for all the reasons that I mentioned. And when we were trying to figure out what it was that we should try and deliver in Parkinson's disease, we settled on this enzyme called glucocerebrosidase, or GCase. And the reason for that were multifold. One is that there's actually a biological reason why we think that this enzyme might actually reduce neurodegeneration and death of those dopamine cells. There's a lot of preclinical studies that suggest this. It's accessible because actually glucocerebrosidase is already an enzyme used to treat a different human disease, Gaucher disease, which you'll probably hear a little bit more about from the patient that will speak later. And because it's being used in humans already, we know that it's safe. And so we opted to try in a very small pilot study, delivering glucocerebrosidase using this focused ultrasound technology. This was a phase 1 trial. Again, going back to the point that this is a safety trial, very early stages, and this was our pilot study done together with the people at Sunnybrook. What we were able to demonstrate is that we could actually deliver this into the brain. So down here is a different kind of picture of a person's brain with Parkinson's disease. We were trying to actually target a very deep structure, which is involved in receiving the brain cells that require dopamine. And so you can see this brightness here shows us that we were able to open up the blood-brain barrier. And all I'm showing in this cartoon is that what we were delivering is this glucocerebrosidase. This cartoon just shows a variety of different ways in which a cell can die in Parkinson's disease. And what we're trying to restore with this glucocerebrosidase is this organelle called lysosome, or the function of this organelle, which is very important in destroying and getting rid of harmful substances, in particular bad proteins that can be harmful to Parkinson's disease brain cells. So the take-home messages from all of this, I hope that you can appreciate, is that our current symptomatic therapies for Parkinson's disease are very effective. So I want you to not take away from this that we don't already have good things for Parkinson's. We do, but we obviously want to do better. Our current therapies do not slow or stop the neurodegeneration. And so we need research, and there is research that's ongoing and expected to accelerate to discover these emerging therapies, not just the ones you heard about, but additional ones as well for people with Parkinson's. And new technologies, I think, are very important for our future because they're going to be important to both improve our delivery, but also the different types of technologies we have for these emerging therapies. And so with that, I thank you very much for your attention. I just wanted to give you a little bit of information here. Our laboratory focuses on trying to discover disease-modifying therapies. If you're interested in any of our work, please visit our website or follow us on Twitter. If you're interested in the Focused Ultrasound trial or other clinical trials for Parkinson's disease, please feel free to email me and I will forward on your email to the appropriate people. We do have other clinical trials going on at the Toronto Western Hospital for Parkinson's disease that are unrelated to what we talked about today. And you can email Suneil about the stem cells trial. And an additional resource is just listed here. This is our movement disorders clinic at the Bloodbrain Institute, where you can visit information about the clinic, but also there is a tab there for research. And you may find that there's other research studies that you'd be interested in participating in. And thanks so much. And we look forward to hearing next from Susan.
Thank you, Doctors Lorraine and Suneil Kalia. I really enjoyed hearing about how the blood-brain barrier can be accessed through ultrasound. It was very interesting. And now we are privileged to hear from Susan. Susan was born in Halifax and grew up in Nova Scotia. She obtained a Bachelor of Arts in Music before graduating from Dalhousie Law School in 1990. She moved to Toronto to article shortly thereafter and has spent her entire legal career in various government roles. She was diagnosed with Gaucher disease in October of 2016 and a short time later noticed her first tremor in February 2017. She was diagnosed with Parkinson's in April 2017 and considers herself extremely lucky to have Dr. Lorraine Kalia as her neurologist and movement disorder specialist. An avid traveler pre-COVID, she's visited 6 of the 7 continents, either on vacation or as a volunteer with Habitat for Humanity, and has an expedition cruise booked for fall of 2022 to Antarctica to complete her bucket list. This past spring, Susan participated in phase 1 of Dr. Kalia's Focused Ultrasound clinical trial. Welcome, Susan.
Hi, everybody. My name is Allison Lee, and I am Parkinson Canada's lead of programs and services. I am so thrilled to have Susan with us here today, taking part in the Focused Ultrasound clinical trials. Welcome, Susan. Can you share a little bit about yourself with us, what you do, what are some of your hobbies?
Thanks so much, Allison. It's really an honor to be here. Well, I tell you a little bit about myself. I was born in Halifax and studied music before I went to law school. About 30 years ago, I moved to Toronto, and I've spent my entire legal career with the government. And as I say, about 30 years ago, because I just celebrated 30 years of service. I think outside of work, my number one hobby is to travel. I'm an avid world traveler pre-COVID, both as a vacationer and as a volunteer for Habitat for Humanity. I've been to 6 of 7 continents, and next year I have a trip booked to Antarctica. So hopefully I'll cross that off my bucket list.
That is so exciting. You will be one of the few people in the world that can say that you went to Antarctica, because I know it's not that many. Susan, can you share with us when you were diagnosed with Parkinson's disease and what that journey has been like?
Yeah, certainly. So it's interesting for me. I was diagnosed first with genetic disorder called Gaucher's disease, and that happened in October of 2016. And then when I was diagnosed with Gaucher's disease, I was told that I was highly at risk for Parkinson's. So I was referred actually to Dr. Kalia just as a sort of an introduction, because she has an interest in Gaucher's disease patients who have Parkinson's. At that time, I had no Parkinson's symptoms. But a couple of months later, I developed a tremor in my right leg. So I fairly quickly deduced that I had Parkinson's. Interestingly enough, I had also lost my sense of smell, or started to lose my sense of smell, probably 10 years ago. So when I was diagnosed with the genetic disorder and I looked up sort of some of the symptoms of Parkinson's, because I frankly didn't know that much about it, I read a little bit about some people losing their sense of smell. And then, of course, when I developed the tremor, I was pretty sure that I had Parkinson's. So I was formally diagnosed in April of 2017 with Parkinson's, and I became a patient of Dr. Kalia's. So again, I think I'm fairly unique because a number of people that I've met over the last 4.5 years have taken a long time to be diagnosed, have had lots of other symptoms, sort of struggled to find, what was wrong with them. I saw a neurologist who said, your symptoms are consistent with Parkinson's. Do you have any questions? And I said, no. I think I'm probably one of the few people who went, expecting a diagnosis. So I think in that respect, I feel very, very lucky because I was diagnosed very quickly, and I got very quickly in to see a movement disorder specialist. And so fairly, went on meds fairly quickly. The medication has helped considerably. And I'm sort of living with Parkinson's. And the other thing about my diagnosis was, in terms of the connections and sort of the Parkinson's piece, is that I was referred to a Parkinson's physiotherapist specialist. And through that, I got connected to the Pedaling for Parkinson's and the Rigid Riders, and through that, a support group, and through that, another support group. And so I feel, again, sort of a confluence of events led me to where I am, and in a good place, as good as I can be with my Parkinson's.
Yeah, absolutely. And that support circle around, right? Like how you were connected from one thing to another, and then it keeps on going. That's pretty incredible.
Yeah, and I'm not somebody who reaches out. I think, and I've sort of heard this repeatedly, that again, sort of that Parkinson's can be a very lonely disease. And so it wasn't in my nature to kind of reach out to support groups. But I have to tell you, I can't imagine now not spending my time with these groups. We had a meeting yesterday. There's another meeting tomorrow night. So, there's lots of support out there, and it comes in different forms at different times. But it's super helpful. And the key thing, I think, is community, which I think, maybe I'll talk a little bit more about why I became involved, but community is incredibly important. And so, we're not alone, and everything we do affects other people. And again, that's another piece for me why research is so important, because what I do can influence or impact, not only my colleagues now, but people in the future. So.
Absolutely. And I know in that process, you touched on that research piece there. Can you share with us, not just getting involved in support groups and community that way, but what piqued your interest or what motivated you to start volunteering in clinical trials?
Well, so Dr. Kalia approached me because she said that there was this process with respect to the focused ultrasounds, and that they were looking at doing a phase 1 clinical study that involved opening the blood-brain barrier and administering a particular drug. And what was particularly interesting to me was this is the drug that's used to treat Gaucher's disease. Now, I don't get that treatment because you need to be medically, it needs to be medically necessary. It's very expensive. It's very time consuming. And I'm not ill with Gaucher's disease. Again, I'm pretty lucky that way. But so it was particularly targeted, I thought, to me. Dr. Kalia laughs because I call myself a unicorn, because Gaucher's is rare, and Gaucher's with Parkinson's is even more rare. And so for me, I was, in my own mind at least, I was an ideal candidate for this study because of the fact that it was studying people with Parkinson's and using this particular drug. So Dr. Kalia spoke to me first. I remember that she said to me, I said, that sounds great. I'm totally interested. And she said, well, you'll have to shave your head. And I said, that's fine with me. That's not a problem. So Dr. Kalia sort of first approached me, and then I got a call from the clinical study coordinator at the hospital, who spent quite a bit of time with me, sort of talking to me about what it would look like, and the various tests involved. It was a fairly lengthy process, because a lot of people in their earlier studies using the focused ultrasound they found that a lot of patients got excluded because of various tests that they did to ensure that they were medically stable, or, whatever the criteria were. Behind the scenes, I'm not sure. So I had MRIs, I had CAT scans, I had EKG, I had a spinal tap. One of the very last things they did was the spinal tap. And fortunately for me, I was continuing to be eligible. And I will say that part of the process that I was particularly impressed with was all the time, every time I went, it was very patient focused. And it was very clear to me from the very beginning that at any time I could back out, I could change my mind if I got frightened, if something happened that I changed in my life. Right up to the time of going into the MRI machine, even having the head frame on and my head shaved, I could change my mind. I mean, that gave me a lot of comfort to know that, that would be okay and that everybody would accept that. So for me, it was sort of I hate to say it was a no-brainer, but I'm very, very, again, I keep saying I'm lucky, but I'm very lucky. I'm single. I don't have any children. I have a supportive family. I have a supportive employer. And so a long time ago, somebody said to me, if not you, who? And by that, she meant, like, if you don't step up to the plate, who's going to do it? Somebody asked me in one of my support groups why I was doing this, if there was going to be no benefit to me. And I said, I'm doing it for humanity. And I thought afterwards, that sounds really pretentious, but it's true. We need to develop a sense of responsibility to everybody else in the world. It's not just about me. It's about what can I do for the people around me? And for me, that's the people around me with Parkinson's. And it's the future generations of people who, knock on wood, won't have Parkinson's, or will maybe get Parkinson's, but there'll be a cure. So I feel very strongly that sense of sort of responsibility to try to help where I can. So it really wasn't much of a choice in terms of saying yes. I fairly quickly said yes. And as I say, I feel incredibly lucky to have been part of the process.
Absolutely. And that's, I think, quite incredible to hear your experience, even of how patient-centered it is. What would your hope be for the future of Parkinson's research? And what would you share with others if you were able to encourage them to do so from your experience?
I would say, one, reach out. I mean, I was lucky because my neurologist asked me, but I've since been a bit more of a interested in looking at the clinical website and seeing what's available. And two, I would encourage people to register with the Canadian Open Parkinson Network. I think that, for me, I personally benefited from the fact that there was knowledge that there was a connection between Gaucher's and Parkinson's. And that only happened because people did research and people shared information. And so I'm a direct recipient of at least having some better path on my journey, because people before me, sort of gave their DNA, allowed their information to be shared. My mother had ALS. So I, again, have a personal connection to, research helping, in terms of any kind of neurological disease. But I particularly think with Parkinson's, there's so many variables and there's so many different experiences that people have had that there's just a wealth of information, and there's so many ways that patients can help in terms of research, or, as I say, becoming a member of the COPN, that I think that there's every chance that there's going to be a cure, maybe not in my lifetime, but fairly soon. And it gives me hope. So that's what I would say.
Thank you so much for sharing, Susan. It was so lovely to hear from your experience, and we are so thankful to have you here with us today.
Thanks again, Allison. It was really lovely to be here.
Thank you so much, Susan, for sharing your story with us. It was very inspiring, and I'm amazed at how brave you were to be such a pioneer. Thank you to all of our speakers today, and we now have some time to answer some questions. The first one was submitted upon registration. It says, given the limited number of neurologists in Canada, how readily available will these emerging technologies be to Parkinson's patients? How can patients access these treatments?
Thanks for the question, and I think a really important one in terms of access, which is a struggle, admittedly, across the country. So with respect to access to these therapies, I think we first need to make sure that they are going to be therapies that are worthy of access. As we're trying to emphasize, we're in the early stages of development and discovery for these particular therapies, and there are even ones that are earlier in development. And so phase 1 trials are those first stages of trials where we try to ensure for ourselves and for people with the disease that it's safe to proceed with further study. And some of these emerging therapies will fail at that point because they're just not safe enough. But brave people like Susan, who participated in the phase 1 trial, are people who help us to move further. And after that, we move on to what we call efficacy trials, where we're really not just, we feel that the medication is safe, but now we're really trying to see if it does what we want it to do, which is stop Parkinson's disease in its tracks. And so we're still, these are years away in terms of having answers to those things. And so the expectation is when we start seeing a signal that these therapies will be beneficial, that at the same time, obviously, we would need investment and partnerships with industry, who would be the kind of drivers to ensure that there is access to these therapies. From the focused ultrasound point of view, we've partnered with the people at Sunnybrook because that's their expertise. So I can only speak briefly to what I hear is in development there. Right now, the way that it's being delivered, and Susan can speak to this firsthand, is going to Sunnybrook, having her head shaved, putting the helmet on, going into the MRI. But the future for that technology is actually evolving as well, where physicists and engineers are trying to develop ways where that technology could actually be done in a clinic. And so instead of having to, go to a single center, you could go to a, similar to how people who receive chemotherapy go to an infusion clinic, you would go actually to a clinic, put on a helmet, sit in your chair, and have this kind of therapy delivered. And so that kind of technology is being developed, I think, alongside. And so, so that's going to obviously improve access to this therapy, should it become something that becomes part of our arsenal for treating Parkinson's disease.
Thank you. There was a follow-up question that was submitted online today asking about how long do you think it takes to get through phases 2 through 4 for the trials and to be able to get it delivered to the public?
So that's very variable from trial to trial. It's also very variable depending on what kind of who's investing in that, how motivated industry is, who are usually the people who are supporting these trials. I can tell you the focused ultrasound trial we did was what we call investigator initiated, meaning it was us as people who research Parkinson's disease and the Sunnybrook people who research focused ultrasound who brought this forward and looked for funding elsewhere. And unfortunately, there's not a huge amount of funding for that kind of work. We've been fortunate to be able to get funding from the Western Brain Institute to do a follow-up study to the one that we're doing. But a huge amount of this kind of investment actually comes from industry. And so this is where you as a community, us as a community, we have to be advocating so that industry sees neurodegenerative disease and Parkinson's disease as something very much worthwhile investing in.
Great, thank you.
And I was just going to add, the pipelines are different depending on the therapy and the technology. So taking something from the lab, the basic science lab, to the human brain may take 10 or 15 years. New technologies can be turned around from the lab to patient trials within 5 years. And then other strategies, like taking existing drugs for other diseases that we already know are safe and approved, similar to a strategy that was done in our focused ultrasound study, and move it to a different application. We call that repurposing. The timelines for repurposing drugs is also much shorter. So the way we look at it, we need to have investments in all 3 pockets and have different timelines for completing the studies to get things that are effective and safe to patients as quickly as possible.
Thank you. We've had a lot of questions submitted that are the same, asking about if there's any age limits on access to the ultrasound therapy.
Yes, so there are the good and the bad of clinical trials is that they try and be quite uniform in some scenarios, obviously try and be safe as well. And unfortunately, when people are looking at safety, they often look at age as a consideration because as people get older, comorbidities are often associated, and it's not uncommon that there might be a hard cutoff for an age that is in excess of what is felt to be safe. From the point of view of the focused ultrasound trial, I would encourage people who are interested to reach out, and then we can determine whether or not the person is eligible or not. There are other caveats, not just age, that would prevent somebody from being a candidate. So we don't want to have people who are at increased risk of bleeding, for example, because there's the potential of that, and a variety of other things. And so I think usually what I discuss with people when we talk about clinical trials is I think the first thing is, are you interested? Are you the type of person who wants to be involved in the clinical trial? And then we look to see if you are eligible. And so I think if anybody has some interest, I would first explore whether or not you're eligible before feeling like not moving forward with at least inquiring.
That's some great advice. Thank you very much. We've got a question now for Susan. Did you have any fears about anything before participating in the Focused Ultrasound trial?
So interestingly enough, I was thinking about this very question. I thought someone might ask this question. And the truthful answer is no. I think I'm the kind of person that once I've decided I'm going to do something, I try to then not let the fear take over. So I felt quite comfortable, as I said before on my taped interview, that I could change my mind at any time. And I think even just knowing that gave me the comfort of feeling like I was being safe. It was pretty clear that the team around me was very experienced in using the focused ultrasound. And so I wasn't really afraid. It's weird, I know, perhaps. I think people think I should have been, but I wasn't.
Great, thank you so much. And we've got one more question regarding the blood-brain barrier. The question is, does dissolving the blood-brain barrier potentially expose the brain to other agents such as viruses?
Yeah, so the blood-brain barrier is there for a reason. And one of the reasons very much is to protect our brain from things such as viruses. The advantage of this technology is that it opens up the blood-brain barrier, but only very temporarily. And so Susan explained to you many of these tests that she had done as part of the clinical trial. And one of them actually was to image her kind of before things were opened, and then to image her brain again after, and to prove to ourselves that that blood-brain barrier closed, which had previously been shown in other people and in patients with other neurological conditions. So the expectation would be that during that time when a person's treated, because it's just a temporary time, that we obviously wouldn't want to be treating somebody who was ill or sick, and that's part of the consideration with the treatment. But we know that we all need the blood-brain barrier for safety, and so it's this very temporary opening, like less than a day, hours, that it opens up and then closes back up again and goes back to its important job of protecting the brain.
That's great to know. Thank you very much. Well, we've run out of time for question and answers, but I wanted to thank everyone for attending today's session, and a very special thank you to today's presenters. Like me, I hope you found the information very valuable. All of the resources that were mentioned in today's presentation and in the chat window, along with a link to a recording of this presentation, will be included in a follow-up email that we will send to you. Our December webinar is going to be on speech and swallowing challenges, and it takes place on December 9th. Watch for the invitation that we'll be emailing you towards the end of this month. Thank you so much for joining us today, and have a wonderful rest of your day.